Today's explainer for the Simplified Hub platform is all about breaking down the absolutely fascinating process of mitosis. Now, to really grasp how a cell perfectly replicates itself, I actually want you to step away from biology for a second. Think about construction. We're going to visualize exactly how a cell builds a brand new identical house right next door.
Okay, so let's quickly run through our syllabus for today's build. We'll start with introducing cell division, then move into interphase, prophase, metaphase, anaphase, and telophase, and finally we'll wrap up with cytokinesis.
Let's jump right into Section 1, Introducing Cell Division.
So, what is the ultimate goal of our little construction project here? Well, mitosis is simply the process of cell division, where one single cell divides to produce two genetically identical copies of itself. We aren't just building a house that kind of looks like the original. No, we are building an exact, down to the millimeter replica.
Before we break ground, let's cover our main learning objectives. By the end of this explainer, you should be able to define mitosis, name every single phase in the exact right order, and, this is absolutely crucial, explain exactly what's happening to our building materials. I want you to keep your eyes locked on the chromosomes. They are literally the lumber, the steel, and the bricks of our cellular job site.
All right, moving on to Section 2, Interphase. Now here's a stat that always surprises people. A cell actually spends the vast majority of its life right here in interphase. It makes sense, right? You wouldn't just start pouring concrete without blueprints and permits. A cell spends most of its time doing vital prep work, because you really can't build a second house without laying down the proper groundwork first.
So, what exactly does that groundwork look like? Well, first, the DNA inside the chromosomes copies itself. Basically, we're duplicating our blueprints. Then, to get ready for the big move, the chromosomes actually become thicker and start to coil up really tightly. Think of this as bundling all your lumber and steel beams tightly together so they're way easier to transport across the job site.
Which brings us to Section 3,
Prophase.
To really understand prophase, you need to meet your on-site managers, the centrioles. These are cylindrical organelles that divide and move apart, forming what we call asters. Now, why do we care about asters? Because they're basically the cranes and rigging that will help organize all of our building materials later on. They're the stagehands moving all the heavy equipment into place.
You can really see the rapid teardown happening here. In early prophase, the nuclear membrane starts to disintegrate. It's kind of like taking down the fences around a property line. Then, by late prophase, the asters have moved to completely opposite poles of the site. The membrane is totally gone, and our bundled building materials, the chromosomes, are just sitting out in the open, ready to be moved.
Okay, Section 4, Metaphase. Let's take a quick anatomical pit stop for your notes, class. The centromere is the very center of a chromosome. I want you to imagine it as this heavy-duty metal bracket or strapping that's holding our bundled pair of building materials together. And trust me, this bracket is going to take a lot of tension in just a moment.
Because here is where the real heavy lifting happens. These microscopic protein structures called spindle fibers act exactly like steel crane cables. They reach out, hook right onto those centromere brackets, and just pull. They physically tug the chromosomes until they are forced to line up perfectly straight down the middle. That line? That's the exact property line of our two new cellular lots.
Moving right along to Section 5, Anaphase and Telophase.
Since things are literally pulling apart now, let's update our terminology. Once that bundled chromosome is actually divided, we call the resulting halves chromatids. The materials are being actively separated so that each new house gets exactly what it needs.
Boom! Anaphase hits. Those centromere brackets suddenly snap under the intense tension. Half of our unbundled materials, the chromatids, are dragged away to one side of the lots, and the other half get pulled to the complete opposite side. It's a very forceful separation. And at the exact same time, the outer cell membrane itself begins to pinch inward, basically digging the trench between the two properties.
As we roll right into telophase, that trench deepens. The cell membrane constricts even further. And inside, forming around these two newly separated piles of building materials, two completely separate nuclear membranes begin to form. We are literally putting up the walls and the roof on two separate houses at the exact same time.
Finally, Section 6, Cytokinesis.
This is it, the grand finale. The cell membrane pinches inward completely until it just snaps shut. Mitosis is complete. Where there was once just one lot, we have successfully subdivided into two genetically identical daughter cells, and each one is fully stocked with the exact same building materials we started with way back in interphase.
Let's quickly review the blueprints to see that complete sequence one more time. We've got interphase for prep, prophase to clear the lot, metaphase to line up all the materials, anaphase to forcefully pull them apart, telophase to build the new walls, and cytokinesis for the final property split. It's just a totally elegant repeating loop of construction.
But before I dismiss you all, I want you to consider a quick reality check from our source material. The simple diagrams we use usually show, just a couple of chromosomes floating around. But a human cell undergoing this chaotic build is actually wrangling 23 pairs of chromosomes at once. Imagine having to manage 46 massive bundles of steel and lumber simultaneously, coordinating all of that without a single mistake. The coordination is honestly staggering.
Which brings us to the end of today's lecture. I want to leave you with this final thought. Considering the sheer, unimaginable complexity of moving 23 pairs of materials through this chaotic yet perfectly timed construction process, how many millions of times is this happening in your body right at this very second? It really gives you a whole new appreciation for the microscopic architecture of life, doesn't it? Thank you so much for your attention today, class, and as always, keep learning.